Thank you, everyone. I'm really pleased to be here to give you an update on the work we're doing at ViaCyte. ViaCyte is a privately held San Diego-based company, and I'm going to give you an overview of what we're doing today, starting with some key messages. And the real headline message on ViaCyte, we're a company that's been around about 10 years. We have actually more than a decade. We have been working throughout that time on cell replacement therapy, and the focus has been on type 1 diabetes. So the headline is we are developing what we consider a functional cure for type 1 diabetes, and that product is in the clinic. It also has potential applications for insulin-dependent type 2, but there it would be a treatment, not a cure. So the technology we're based on is a stem cell-derived islet replacement therapy. It's important to note that we don't actually treat patients with stem cells. We start with stem cells in the manufacturing process. We manufacture up to a pancreatic progenitor cell, and we implant that. And part of the technology is the implantation technology, a device. So this is a combination product of a biologic, the cell therapy, and the device. And we also have work going on in the company on gene editing for making cells more immune evasive, but that's a more long-term project. We have two products at the clinical stage. One we call PEC-Direct. It's what we expect to be the first generation product that is targeting patients with high-risk type 1 diabetes. And then a second generation product called PEC-Encap that is designed to treat all patients with insulin-dependent diabetes. The competitive advantages, we have been in this area for a long time. Our largest single competitor in this field was a division of J&J. We consolidated that division called Betalogics with us about 1.5 years ago. So between the two of us, we've been working in this space and really have dominated the field, both on know-how, especially on intellectual property. We have, between the two organizations now, the combined patent portfolio is over 500 issued patents throughout the world, over 100 issued in the U.S. And the commercial opportunity, we are the first to the clinic. We are venture capital financed, but about half of our financing, we've raised about $180 million since inception. About half of that has come from patient advocacy groups. We have had over $70 million come in from the California Institute of Regenerative Medicine, about $15 million from JDRF. We are looking to raise some additional capital at this time. So the idea of essentially curing a patient with type 1 diabetes is not new. Back since around 2000, there was a protocol developed called the Edmonton Protocol using cadaver islets. And these cadaver islets were designed to replace the cells that are lost, the beta cells that are lost to this autoimmune disease. And about 1,500 to 2,000 patients have been treated today. Most of these patients are still insulin independent after 5 years. In fact, one of the first patients who received this is still insulin independent, cured 18 years later. It's a very effective treatment, but it requires anywhere from 1 to 3 pancreases to treat a patient, and there's just a very limited supply of pancreases to make these islets. So what we're doing is essentially designing a way to produce an unlimited supply of cells for this cell therapy. This slide summarizes almost a decade of work, but very quickly tells how we do this. We basically start with a stem cell bank. It's a stem cell line, an embryonic stem cell line that we isolated about 10 years ago called Site 49. It's probably one of the most studied and tested stem cell lines in existence. It's been through multiple studies, GLP safety studies. It's been reviewed by regulators in the US and Canada and now being reviewed in Europe and gotten the green light. So we use that. We have those banked. We have working master and working cell banks. We take a vial of cells from a working cell bank. We then scale that from millions of cells to billions of cells and then go through a 12-day differentiation process to bring these up to what we call PEC-01 cells, which are pancreatic progenitors, which are destined to go on after in vivo implantation to perform all of the elements of an islet. And in order to deliver this, we then load that into a device, and that device is implanted subcutaneously into the patient. And what you can see at the bottom of the slide, this is mouse data, but when the cells mature in the subject, you get a full complement, almost identical to an islet of insulin, glucagon, somatostatin, producing all the various factors. It is a key component is how to deliver it. We have two delivery technologies that make up the two products, the first and second generation. So the PEC-Direct product, which I said was the first generation, uses a device. You can think of it like a tea bag-like device, but this is a tea bag that has had ports engineered into the surface of that tea bag, and that allows direct vascularization. That makes for a very robust implantment. It's impervious to foreign body response. And it's very similar to what's seen with the cadaver islet transplants, but it does require immune suppression, which is why this first generation product is for the high-risk type 1 diabetics. The second generation product we call PEC-Encap. It's also been in the clinic. It uses that tea bag, but now the tea bag is designed such that it prevents any cell-cell interaction. And in this way, it protects those cells, those allogeneic cells, from immune rejection. However, you do need to get a vascular network formed on the surface of that membrane, and so it makes it much more sensitive to foreign body response. And we knew, I'll get to that in a minute, but we've looked at both of these products. They both deliver the same cells. Whichever one you look at in various animal models, it's very effective. It'll control blood glucose at or near the human set point, and you can see that in this mouse here. If you look at that green line, that's a normal mouse that's been implanted with our cells, and over time it will go from the normal mouse blood glucose level, about 150 milligrams per deciliter, down to the human level of about 70 to 100, and it will remain that way for the life of the mouse, or in this case until we pull it back out, and then it will revert back up. So it's very effective at controlling the blood glucose. It does it in a very insulin responsive manner, or glucose responsive manner, and we've never seen any evidence for any hypoglycemia. However, one of the things we knew when we were developing this was that we were going to have to deal with the foreign body response to the device. PEC-Direct isn't impacted by that, but PEC-Encap definitely is. And we knew from different animal models where we had models where it wasn't such an aggressive foreign body response where it worked really well, to models where we had very aggressive foreign body response where it didn't work as effectively. And the question was, where does human stand? So we basically started by taking PEC-Encap into the clinic at subtherapeutic doses to really answer that question. And this slide summarizes data from 19 patients that have been studied in the clinic with PEC-Encap. And we found it was safe and well tolerated. We had very good evidence that the encaptor drug delivery system, this encapsulation technology, was protecting against the adaptive immune system. We saw no allogeneic, no autoimmune rejection, or even sensitization. So that was good news. And as you can see in these first two panels, we did see when the cells got well vascularized in the device that the cells did differentiate, became islet-like tissue as we expected. However, what we did find was that most of the time we did see a very aggressive foreign body response that was limiting the ability of the cells to mature in all parts of the device. And therefore, we feel we have more work to do on this, and we really verified that we are more like the more aggressive rat model than we were the mouse model. So with that, we move forward with PEC-Direct, and the reason is shown on this slide. These two top shows the rat and the human that I just showed you, showing that foreign body response. All those yellow arrows are pointing to foreign body giant cells that are forming a contiguous layer on the surface of this device. But with PEC-Direct, you still get that foreign body cell layer on the device, but because you have these ports that allow direct vascularization, it has no impact on the implant. The implant remains very healthy, it proliferates, it differentiates, and it will control the blood glucose. So we knew PEC-Direct didn't prevent the foreign body response, but basically worked around it. So we've moved forward with PEC-Direct into the clinic. We filed an IND and a Canadian CTA towards the beginning of this year. We started a phase 1/2 trial. The first cohort is the phase 1 portion, if you will, where we gave subtherapeutic doses, and we had planned to do at least 3, but up to 15 patients in cohort 1. We have now done 5 and decided that's sufficient, at least at this point. And what we put in the first cohort are what we call sentinels. They're the mouse-sized devices. We put 6 to 10 of these in at various locations. We can put them in the flank, in the forearm, and then we pull those out periodically to understand how the cells are doing, to do histology on them. But we are now going into the phase 2 portion of this, where we give a fully therapeutic dose of the cells. We're giving enough cells, we believe, to cure the patient, in fact, about 2x what we feel is necessary. And the efficacy endpoint is very clear. These patients, when they're enrolled into the study, are what's called C-peptide negative. C-peptide is a biomarker for insulin. And so they're true type 1 diabetics. They're not able to produce any insulin on their own. And if after we treat them with these cells, they're now producing C-peptide at therapeutic levels, then the only explanation is it's from our product. So it's a very clear and concise efficacy endpoint. We're also looking at insulin utilization, hypoglycemic events, A1C, many other endpoints. But you only need about 36 to 40 subjects to definitively show efficacy because of that endpoint. So that's what we're doing in this phase of the study. This is just a little bit of data, a teaser, if you will. This is from the first 2 patients in cohort 1, where we had pulled sentinels out at 4 weeks. And what it's showing is we really accomplished what we wanted to accomplish with PEC-Direct initially, which is getting that direct vascularization. You can see in these slides, it's a little hard to understand maybe what's going on there. Those circles are from a structural mesh that's over the surface of the membrane, but you can see kind of the membrane, and on each edge is the weld, and you see it's full of cells from end to end, and there's evidence we can stain for blood vessels and such, and we can show that direct vascularization. So we're off to a good start. This is still early. We'll be pulling sentinels at 8, 12, and 16 weeks from these patients, and of course looking for efficacy in the second patient group. So this shows our timeline. I won't go through this in any detail. I'll just say that some of the key milestones coming up for us. Around the end of this year, we'll have more sentinel data from these first few patients that will give us a good understanding of how the cells are doing and how they're maturing in the patient. But importantly, of course, is this phase 2 portion. By about mid-next year, we expect to have the first group of patients with efficacy endpoints evaluated, and that will give us our first preliminary evidence of efficacy. We plan to use that to file for breakthrough therapy or RMAT designation. We're actually in discussions with the FDA now on that. And then the third milestone is by the end of the year, we expect to have all patients enrolled. We evaluate efficacy after 6 months after implant, so that would put us out into early 2019 to have that definitive efficacy. We plan then to have an end of phase 2 meeting with the FDA, and assuming we have the breakthrough therapy or RMAT designation, we would propose essentially flipping this into a phase 3 registration trial by adding additional patients, probably somewhere in the neighborhood of about 100, 150 patients total. So PEC-Direct advantages, we believe it has a low technical risk because of the proven efficacy with cadaver islet transplants, the results with the PEC-Encap clinical development showing that we're on the right track, and the fact that we've shown there's no impact of the foreign body. We expect the accelerated and cost-effective development, and even though we're talking about a subset of patients, about 10% of type 1 diabetics, we brought Navigate in to do a full-blown commercial assessment of this, and they pegged it at for the U.S. alone, about a $1.6 billion peak revenue opportunity. Obviously, that's much smaller than the revenue opportunity for treating all type 1 diabetics and insulin-dependent type 2s, but it's still substantial for the early company. So we are aggressively pursuing the development of PEC-Direct and going forward with that product, but we certainly have not taken our eye off of expanding this to all patients. We're continuing to work on PEC-Encap. The material that we use for the membrane that's the active component of the device is expanded PTFE. It's essentially Gore-Tex. And we have a collaboration with W.L. Gore, the world's leader in this technology. About 40% of Gore's revenue is generated from implantable medical devices, so they have a lot of expertise in this area and a lot of expertise for reducing that foreign body response. So I think we're making excellent progress on that. We've kept the IND open on PEC-Encap and can move very rapidly back into the clinic as we improve the device. We also have another project we call PEC-QT, which is using gene editing technology to reduce the immune profile of these cells, these pluripotent cells. That would then give us another way to treat type 1 diabetics, but also open that very well-studied stem cell bank to be used for other applications. So with that, I think I'm out of time, so I'll stop there.